Okay, so let's talk about how a virus is going to grow inside of a host. So, for a virus to be able to multiply, it can't just sit around by itself. It's going to have to be able to invade a cell and then take over this host metabolic machinery. So, if you're looking at how many viruses are there, there might be a bunch of them that then, when they start infecting a cell, you'll see them start to disappear from the media because they're all getting into the cell. And then they start growing and multiplying and being released. So you see more virions being released. You see a lot of virions and a lot of virions. And then if this is an acute infection, it's eventually going to be stopped. And the numbers are going to go back down as the person recovers. A bacteriophage is a virus that infects a bacteria. It might be able to target the cell wall, the fimbriae, or the flagella of the bacteria. And they can be used in a clinical setting as a treatment to treat bacterial infections. So phage therapy would be taking a pill made of living viruses to kill off that bacterial infection. It's not a very it's not the first thing you're going to turn to treat an infection, because people are going to be a little bit skeezed out sometimes if you say, hey, you're sick, here's some viruses, why don't you eat this? There's maybe some marketing that needs to go on there, but a lot of times people who have very dangerous infections that are antibiotic resistant and they get desperate to do anything, that's kind of where phage therapy is at this point. A lot of experiments and a lot of use going on. I think phage therapy has a lot of promise and potential, although right now we're not at a place where it's a common therapy. Then a virus, once it gets into a cell, there are two different types of cycles it could go through. A lytic cycle is when the phage infects the cell, grows, makes a bunch of itself, and busts out of there and spreads to the next cell. So the cell is going to be killed by the end of this cycle. A lysogenic cycle is when the cell stays alive. The phage is going to incorporate its DNA into the host DNA. So now you have kind of this recombinant DNA that's part bacterial DNA, part host DNA. That's referred to as a prophage. And once you have phage DNA in the cell, there's new genes in that cell. So you'll see phage conversion. This is when the host cell exhibits new properties. And it's not only that you now have new virus genes in it, but that virus gene could have inserted itself in the middle of another gene. So it could have just broken apart a gene that was doing something. It could activate a gene that shouldn't be activated at that point. It could turn off a gene that shouldn't be turned off at that point. So that's all involved in conversion, where that host cell is starting to have new properties. So for a bacteriophage to get in through the lytic cycle, first we have what's called attachment, when the virus attaches to the cell. Then penetration, when the viral DNA is injected into the cell. Then biosynthesis, as the virus starts making lots of little pieces. It makes its new tail fibers, it makes new capsids, it makes new DNA. It'll assemble them together and mature into creating virions. And when those virions are formed, it will release. It will lyse the cell, that means bust out that cell, break it apart, and explode and spread to the next cell. So that's a lytic cycle here. The DNA gets in, makes more viruses, busts out, spreads, injects itself, makes new viruses, spreads. That's a lytic cycle. Short name, short cycle. Lysogenic name is a little bit longer name, a little bit longer cycle. So the phage attaches and injects its DNA. The DNA is here. Instead of just going on and growing more viruses, it sticks its DNA into it. So now you have a prophage that has the purple DNA from the bacteria and the pink DNA from the phage. Then every time the cell divides, it's going to divide all of its DNA. So every time the cell is dividing, it's building more and more copies of that phage DNA. So you're getting lots of cells now that have that phage DNA in it, just because the cell is dividing again and again and again. Eventually something can trigger the cell to excise that prophage DNA, and that cell can go into the lytic cycle, where it starts maturing virions and releasing and causing more cells to be infected. So that cycle we just went through for a virus to multiply: attachment, when the virus is attached to the cell; penetration, when it penetrates in. This could either be endocytosis, where the whole virus is taken in, like what you see here, or by fusion, or just we just saw in the bacteriophage where only the DNA is penetrated. Then uncoating, when everything's taken apart. Biosynthesis, where everything's getting made. Maturation, where all those pieces are now forming virions. And then release. Release can happen by budding. So here, this enveloped virus, as it is going to be released, it's going to take some of the cell membrane with it. That's budding. Here's a little bit more picture showing budding. It's just going to start putting its own proteins into the cell wall or into the cell membrane and kind of push its way out. It could also escape by rupture, which is what we saw with the bacteriophage. Human viruses also can escape from cells by rupture, where it just breaks apart the cell. So how do bacteriophages get nucleotides and amino acids if they don't have metabolic enzymes? Describe the lytic and lysogenic cycles of viral multiplication, and describe the events of attachment, entry, uncoating, biosynthesis, maturation, and release of a DNA containing viruses. Okay, so go through those questions. Make sure you can answer them. Let me know if you have any questions, and then I'll see you in our next video. We'll finish up chapter 13. Bye.